My name is Crystal Bittis, and I am an MD PhD student here in the Gibbons Lab at the ISB. I am sharing the last, but certainly not least session that will focus on the endocrine effects on the gut microbiome and its associated metabolites. This session is going to feature two fantastic talks, a Matthew doubleheader, first by Dr. Matthew Carter, followed by Dr. Matthew Redenbaugh in the second half of the session. So to start, it's my great pleasure to introduce Dr. Matthew Carter, who is a postdoctoral fellow at the Arc Institute and Stanford University, working with Professor Christoph Theis. He earned his PhD in microbiology and immunology at Stanford University with Justin Sonnenberg, where his research focused on diet, microbiome, host interactions. His work combines clinical dietary interventions, multi-omics, and gnotobiotic mouse models to uncover mechanistic links between the gut microbiome and host metabolism. Dr. Carter, the floor is yours.
Amazing. Thanks, Crystal. Awesome. I'm very excited to be here. Thank you so much to the organizers for the invitation, especially for someone like myself who is still a trainee. Like Crystal mentioned, I'm currently a postdoc at the Arc Institute in Christoph Theis's lab. The work I'll be talking about today is from my graduate work in Justin Sonnenberg's lab. When I was in Justin's lab, something that we were very interested in was using human dietary interventions to probe host microbe interactions. Myself, as you know, so many of us here study the microbiome. One of the strongest modulators of the microbiome is diet. And one of the nice things about diet is that it's relatively straightforward to just run human studies where you're doing dietary interventions. And the benefit of that is that you can kind of immediately gain some insights into the impact of different diets on human health and our microbiome, and then it can inform further mechanistic studies in model organisms, where, of course, this is kind of an inversion of the sort of typical model of starting things in mice and then hoping they translate into humans. So the model that we were kind of very interested in was just starting with human studies, and then any insights that we gain from these will hopefully sort of be more informative when we go into these more mechanistic studies. To do these human studies, Justin's lab has had a very long-running collaboration with Professor Christopher Gardner in the Department of Nutrition at Stanford. Over 10 years now, I've been very fortunate to be involved in a couple of these studies. Maybe some of you in the audience have encountered some of these studies. The study that I'll be talking about today is called the Twins Nutrition Study. This was a randomized clinical trial involving 21 pairs of identical twins, where basically for each twin pair, one of them consumed a vegan diet for 8 weeks, and one of them, the other member of the pair, consumed an omnivorous diet for 8 weeks. And along the way, we performed kind of multi-omic profiling on various features of their stool and blood. And then the sort of hope is that we can use these kind of multi-omics associations to then inform follow-up studies in model systems. This paper is currently was posted on medRxiv earlier this year, so I would encourage everyone to read it. So some background on why we're interested in the vegan diet in particular is in part because it is very much increasing in popularity, and there's a lot of interest in understanding potential health effects of the vegan diet, which of course involves the complete abstention of consuming products derived from animals. I think it is noteworthy, though, that when we're doing these intervention studies for particular diets, it is not, of course, to say like which diet is better or worse for you. The whole idea is that we can, by doing more and more of these studies, we can start to learn some of the rules and the mechanisms by which different dietary patterns affect different aspects of host health. And so typically when we do these studies, it's Christopher's group that are the world experts in running these clinical trials. And then along the way, they collect the samples and send them to us. And so typically they do a kind of like first pass analysis and paper. And so these are the results from the paper on their side of things, led by Matt Landry and Kate Ward, where basically it's kind of just like high level, clinical labs. And what we see is even over the course of 8 weeks, we see significant decreases in the vegan group for LDL cholesterol, insulin, and weight. So even just like a short change of diet like this can have some beneficial impacts on health. But now, kind of getting into some of the multi-omics analysis that we did for the study, the first thing that I'll show is the result of the untargeted metabolomics analysis on serum samples that we collected. And it resulted, this dietary change resulted in a pretty large impact on the circulating metabolome, which isn't that surprising. One really nice thing about doing these kind of like unbiased measurements with metabolomics in particular is that you can actually measure biomarkers of, in our case, either meat or plant consumption, where especially on the, in a vegan diet intervention, you're doing these studies of people kind of like out in the wilds, you do want to have some way of knowing beyond what they self-report of like, are they actually sticking to their diet? And indeed, we could actually tell from a lot of the metabolomics data that there were some metabolites, such as this like CMPF, I'll point out in here, which is very abundant in fish, for instance. 3-methylhistidine is a marker of red meat consumption. And we do indeed see very pronounced increases in the omnivorous group in some of these metabolites relative to the vegan group. In addition, there are lots of different fatty acids and amino acid derivatives that we see as different, including a couple of metabolites such as indole propionate and lithocolate that are, in fact, microbial metabolites. So now, pivoting to looking at the microbiome, to analyze the microbiome, we performed shotgun metagenomics on the stool samples from these participants over time. And we can look at both species and metabolic pathways that are differentially enriched in these two diets. And so on the left, we have the differential species abundance. And on the right, we have the functional pathways that are differentially enriched, where I've basically labeled the couple statistically significant hits we had based on their KEGG orthology. And there were, in fact, only a couple pathways that were significantly enriched, which is in some ways kind of nice because it can make it kind of easy to focus on different things. And so the pathway that I'll be focusing on for the rest of the talk is this glycine reductase pathway. This glycine reductase pathway is pretty well conserved across clostridial species of the firmicutes, as well as a few species in the Disulfovibrio class of bacteria, where in particular in our study, it was the species Bilophila wadsworthia, which seemed to be, if you look at the abundance of these species that harbor this pathway, seemed to be the species that changed abundance the most during the vegan arm and didn't really change at all in the omnivorous group. So something about this vegan diet caused the abundance of this species, Bilophila, to decrease. This species is very interesting. It's been actually studied quite a bit in the context of different types of metabolic disease as well as IBD. It's mostly been studied in the context of its unique ability to respire taurine, resulting in hydrogen sulfide production. One of the reasons that I think this is the kind of particularly interesting for us is that, in its name, you can see the Bilophila is a bile-loving species. In humans, the two most common amino acids conjugated to bile acids are taurine and also glycine. So Bilophila has been very well studied for its ability to respire taurine, but not as much with regards to its ability to respire glycine, which might be kind of what we're seeing in our data here. Part of the reason why it was particularly interesting that glycine was a hit both in the metabolomics and the metagenomics analysis was kind of this curious finding where on the right we see the consumption, the amount of glycine consumed in the diet significantly decreased. In fact, on the vegan diet in this study, basically vegans, because they're not eating meat, are consuming much less protein. And so basically, the consumption of every amino acid went down quite a bit. However, glycine was the only amino acid where the abundance in serum on the left here increased significantly. Everything else either kind of stayed the same, which you would expect if our bodies can just kind of make if they're non-essential amino acids, or in certain cases, the abundances of different amino acids decrease because they're essential. We can't make them, and so if we're not getting them in diet, they decrease in circulation. Glycine was the only example where it increased over the course of the study. This is kind of interesting when you go and look at epidemiological data, where here this is basically like a risk reduction plot, basically showing different serum metabolites and how they correlate with the risk of developing diabetes. And so metabolites on the left of this line here are associated with the lower risk, the more you have, and glycine is one of them. This has been shown both in terms of like prognostic risk for diabetes, but also amongst diabetics. Diabetics tend to have lower levels of circulating glycine. And there have also been some interventional studies of administering, basically supplementing glycine in the diet of diabetics, and it does seem to have some beneficial effect. So it might be not only a biomarker, but potentially could be actually involved with the etiology of certain metabolic diseases. And so, indeed, the subjects in our study were not diabetics, and so we couldn't look at that directly. But one thing that we can look at is basically the level of people's fasting insulin, where kind of on the road to diabetes, you see this increasing level of insulin resistance in your body, making more insulin. And so, what we see is even in the cohort we have here, the lower glycine you have in circulation, the higher your fasting insulin. So we're even kind of seeing some hints of this correlation in our data as well. And then if you layer on the finding I talked about earlier with Bilophila, where indeed, if you correlate the abundance of Bilophila wadsworthia in the microbiome with the level of glycine in circulation, you see that there's a negative correlation. So the more Bilophila you have, the less glycine you have in circulation. And indeed, the more Bilophila you have as well, the higher level of fasting insulin that you have. So from these kind of multi-omics findings of profiling these different things, we can kind of build this network of associations in the data. And so the kind of model that we built from this multi-omics associations that we're interested in testing is that, in a, kind of normal circumstance, you're eating glycine, kind of as I alluded to, there are lots of things that the liver will conjugate glycine to, such as bile acids that will then enter the lumen of the intestine. And then there are bacteria such as Bilophila wadsworthia that have this glycine reductase pathway, or I don't think I mentioned explicitly, but basically this pathway ferments glycine to acetate. And then potentially in our data, we're seeing that when you sort of adhere to a vegan diet, and we saw that there was this decrease in the abundance of Bilophila wadsworthia, this results in a reduced flux of glycine through this pathway, resulting in potentially more glycine that could then enter circulation, which might have sort of beneficial knock-on effects, which is sort of what the epidemiological evidence suggests. And so kind of in the second half of the story, as I alluded to in the very beginning, we were very interested in taking these associations we find in the human study and attempting to see if we can demonstrate some of these mechanisms, particularly when it comes to these microbial mechanisms in vitro and in vivo models. And so I'll go through a couple of those experiments quickly. Basically, the first thing that we wanted to show, which just had not been shown before, was that Bilophila wadsworthia can actually use this glycine reductase pathway to ferment glycine. And so the way that we did that was basically using a stable isotope tracing approach and mass spectrometry, where basically the way that this glycine reduction works is it's a form of Stickland metabolism, meaning that it's paired to the oxidation of some other metabolite. In this case, we think it's pyruvate. But basically, we can use a heavy carbon glycine, and we can grow Bilophila wadsworthia in vitro in a minimal media and basically measure both the loss of heavy glycine, this sort of M+1 here, and the accumulation of not only acetate in general, but also specifically acetate that has a heavy carbon in it. And what we see is indeed, when we do this, we do see an accumulation of this heavy acetate in the growth media. And in fact, the majority of glycine that is consumed is converted to heavy acetate. And we also just did a sort of RT qPCR showing that when you add glycine to growth media, you do see activation of this pathway relative to media that does not have glycine in it. And so the next thing we wanted to do was basically test for the effects of Bilophila in vivo and see if maybe it can potentiate this change to circulating glycine, and also what other kind of knock-on metabolic effects might be. And so the tool that we used to do that was an amazing tool developed by Michael Fischbach and Casey Huang, as well as Justin, which is this complex designer community called HCOM, which is basically the largest synthetic community that has been constructed to date, which was very painstaking work from postdoc Alice Cheng in his lab, where basically they remarkably showed that if you individually culture, in this case over 100 strains, combine them and put them in a mouse, they will actually form a stable community. And the reason that this is valuable is that, unlike using just like an endogenous mouse microbiome, it's a lot of microbes specific to mice, but also it varies based on the vendor, what have you. You could use human stool, but of course this varies from person to person. So the idea is that it's like a reproducible tool where you can very specifically control the membership. And so there was another great Cell paper from Michael's group where they actually did some of this preliminary work of looking at what happens to metabolism in the community when you remove individual members. And so I worked with a postdoc in Michael's lab, John Fung, who helped me assemble these communities with and without Bilophila. So there's basically a full HCOM community, and then a full HCOM community delta Bilophila. So you drop Bilophila out, and what you see, and basically what we did was we had two groups of black 6 mice on high-fat diet, and basically observed them measured their microbiome over the course of 4 weeks to look at the stability of this community. And then at the end, we basically looked in their serum and liver, et cetera, to basically assess kind of the metabolic health of the host. It's kind of amazing. Like, it really is just a very stable community, and it's quite specific. Like, it is not the case that when you remove Bilophila from this community, you get a bunch of other kind of epistatic interactions between bacteria. It is quite specific that, of course, there's some minor compositional differences, but really it's just the loss of Bilophila from this community. And so when we ran the experiment for 4 weeks, the results to me were very cool, where when you remove Bilophila from the community, we do indeed see an increase in the level of circulating glycine, mirroring potentially what we saw in the human study. And we saw basically many other features, kind of markers of metabolic health, that were improved upon the removal of Bilophila from the community. So there was both lower body weight, lower cholesterol, sort of improved profiles of like liver enzymes, and also some changes to gene expression in the liver as it relates to oxidative stress. So these results were very exciting. Just to return to the model in conclusion, where we did this human study where we performed this multi-omic profiling over time, comparing vegans and omnivores. We kind of saw these associations as it related to circulating glycine, some changes to the microbiome, And then we did some of these experiments in vitro and in vivo to try to demonstrate that indeed, potentially this Bilophila wadsworthia is kind of has some causal role in the metabolic health of the host. So, just in conclusion, I want to thank Justin, no longer in his lab, but I miss him very much. He was an amazing mentor. And then this project was the close collaboration with Chris Gardner's group and also Michael Fisch's group. So thank you very much.
Great talk. We have a couple of questions in the chat. Let me just pull these up here. Okay. So the first one is, desulfyl Vibrio and Bilophila are both H2S producers, which is very bioactive. Do you have any metadata that suggests a vegan diet reduces the abundance of these bugs and has effects on transit and motility or other outcomes outside of insulin resistance?
Yeah, that's a good question. It was uh, we don't have any measurements of like motility or anything like that. It's a super good question. It was kind of interesting that lots of the previous studies of Bilophila was in the context of the things that the question asker was talking about of like H2S production, more of like an inflammatory profile, whereas at least in our hands it was more uh, metabolic kind of. I do kind of wonder that like it is quite interesting that I'm speculating, but the in mice the bile acid pool is very heavily skewed towards taurine. And so I wonder if you get more of this H2S production because there's just much more taurine. In humans, the bile acid pool is much more skewed toward glycine. And so I wonder if that is kind of why you see this like kind of difference in phenotype.
Yeah, that's really interesting. And I mean, it's also kind of connecting back to our first session where we were, or I think it was the first session, right? Where we were talking about IBS patients and then how diet can also impact them.
Totally.
And the potential for, bloating and how that affects symptomatology.
Definitely.
Very awesome. Our next question, do you think that bacterial metabolites can impact anaplerotic stress and therefore be relevant in common pathogenic pathways related to metabolic syndrome? Uh,
Oh, well, I don't know what anaplerotic stress
I
is.
didn't know either, so I Googled it for you.
Okay.
So it is a metabolic state where the TCA cycle intermediates like oxaloacetate, malate become depleted. And one example could be like cancer cells utilizing it. So I guess here they're asking about bacterial metabolites impacting these TCA metabolites.
Yeah, it's very interesting. It's basically it's something yeah, there was so many directions I would have continued to love to explore, and maybe Justin will continue to do so. But basically, the oxidative state of the host is very interesting. Like it was something that we kind of saw when we were looking at like the liver gene expression of obviously the liver is very important for controlling like redox homeostasis of the host. I don't have a specific answer to the question asker, but it is like a super interesting like direction of the sort of oxidative stress.
Yeah, there's always more questions in science.
Yeah.
Let's see. Uh, so great talk. Have you thought about how variation in microbes that can deconjugate glycine from other host-secreted pathways may influence the flux through these pathways in serum levels?
Yeah, it's a great question because it's very interesting that it is, if you look in the literature, uh, it is quite clear that Bilophila can grow, like in a sense, it grows much better when there is more bile around, but it appears to not have its own like bile salt hydrolase, basically. And in fact, when I tried to monocolonize mice with Bilophila, it wouldn't stick. It needed a partner who could deconjugate bile acids. That I didn't have a chance to show that. And so I think that that's totally spot on. And I think it is. It's just hard to say. Like, I think it kind of seems like if this data supports that Bilophila can grow on either the glycine or the taurine, maybe it like doesn't super matter. But maybe if it's something kind of like what I was saying before, where if there's like more taurine is able to be deconjugated and it skews it more towards like an inflammatory phenotype or what have you, then the specific microbe responsible for deconjugation could be very important for that. It's a really good question.
Yes. And then sort of as a tack on, did you see any metagenomic or metabolic difference between the two groups, particularly in regard with protein intake, since it's the main difference between the two diets?
Definitely. Yeah, it's a very uh this person might have been one of the reviewers of our paper. It was definitely one of the bigger when you do these studies, it's very hard to like perfectly control for macronutrient intake while adhering to whatever the rules are of the different diets. And so it's definitely protein intake across the board was much lower in the vegans. It's kind of why, in a sense, you would like of course, the circulating levels of these amino acids, especially the essential ones, are going to be much lower. That's why kind of why it was very curious that glycine was higher in circulation. Like it's kind of counterintuitive. I mean, it was kind of why that was kind of the hook for focusing on it. But definitely, I think there's probably other very interesting stories of some of the other amino acid derivative metabolites. Even some previous speakers today showed that a lot of these metabolites that affect host health are kind of amino acid derived. So there's definitely just like a huge change in the pools of these metabolites between these two diets.
Alrighty, we have some extra time, so
Sure.
I think we can go ahead and answer a couple more questions. Um, let's see. Apart from diet, are there other modalities that you or others are exploring to delete Bilophila from an intact microbiome?
Yeah, it's kind of a holy grail, right? Of just like being able to do that. That's kind of why, at least for now, in these model systems, like I think this HCOM tool that Michael and Casey Huang put together is like an amazing tool for studying this, especially because it does not always the case, but it seems that most of the time when you remove a given species, you can do it rather specifically. In terms of like some type of therapy for humans, I have no idea. It's definitely I don't know. I'm sure there are lots of people working in things with like phage and what have you, but I don't know of any way to do it specifically right now. Maybe, I mean, maybe in a sense, the answer from the study is you should go vegan for a little bit to get rid of this, but I don't know.
Very fair. Great. Let's see. I think I missed a couple. So right here, I found one that says, using metabolic modeling, could you show that the microbiome's predicted glycine fermentation rate is associated with the fasting insulin?
Oh, very interesting. We weren't able to, I see. So can you like measure flux in the human samples basically? That I think I don't think we have a good way to do that. Or at least like the maybe you could do that. Maybe there's a world where you could do some type of like transcriptomic analysis, but unfortunately, it's just the realities of when you do these studies, the stool is not preserved to save the RNA, it's preserved to save the DNA. So it's a little hard to um, look for that directly. And it is also just the reality oftentimes when you do these studies that um, when you're measuring stool, you're getting maybe some sense of the composition, but you're getting a very different sense of the activity, you know what I mean, of what might be happening compared to much earlier in the GI tract. So it's a really good question, but I don't really think we have a way of doing that in the human data that we have.
Gotcha. We have a couple questions from a viewer who is vegan, so they
Oh,
especially want
great.
to thank you for your work. Um, and so let's see. If they could only choose one, we'll start with C. In your mouse colonization experiments, what is the mechanism of body weight loss in mice without B. wadsworthia? Do they move more? Do they metabolize more or less? If you have any speculations.
Yeah, I'm not really sure, to be honest. It was pretty cool. The of course, there's like experiments you can envision using like metabolic chambers or what have you, but those are very hard to do in a gnotobiotic sense. Like a lot of these more behavioral things are harder to do in like a gnotobiotic isolator. In hindsight, and yeah, I mean, it'd be interesting also to look at like food intake and things like that, but it was just kind of because I had no idea that that would be a finding. I didn't even think about those things. and it's a little harder to do when all the mice are co-housed, but um, I don't really know, to be honest. But I,
It's
it's
a
kind
great
of exciting.
question.
Yeah, yeah.
Yeah. Awesome. Maybe we'll, yeah, just do one more and then maybe we can have a little bit of a break unless Dr. Denbo is ready. All right. So since connective tissue and collagen-rich foods are much higher in glycine, it would be interesting to see if an omnivorous diet enriched in these components would affect the findings. Could you elaborate a little bit more on the composition of the omnivorous diet? Uh,
Yeah.
well, primarily meat or, yeah.
It was, the it's a great question and it's a great observation. And because the question is kind of asking like, could you just take an omnivorous diet and supplement glycine kind of to maybe get the same effect or something? The I don't have a specific sense of how much like collagen might be in the diet. It was a quite varied diet, and I would say that it is definitely quite a healthy diet for whatever that's worth. Like it was something I would be very happy to eat myself. It wasn't like some type of sham diet where we're just giving people like cheeseburgers. But yeah, so it was definitely like there was definitely red meat, so there was definitely like some collagen in there. There's fish, eggs. Um, I don't have a specific sense of uh, collagen, but it was on the omnivorous diet, at least in terms of like our ability to track diet and then integrate that with like nutrition databases. The glycine composition of the omnivorous diet was like pretty level from baseline. So it wasn't like people's glycine consumption changed that much.
Great talk, Dr. Carter. And then we'll go ahead and move to our next talk.